Hello there, thank you so much for joining my YouTube channel today. And just like to introduce myself as Miss Malik. If you don't know who I am a GCSE science teacher and also a tutor, and I have been teaching from 2013. I do have lots of experience in GCSE chemistry, biology and physics. Now today's YouTube video is going to be based on my GCSE predictions for Biology Paper 1 that is going to take place on Tuesday the 13th of May. Now, I do advise you to learn all topics and not to just rely on predictions, because predictions may or may not come up. This video today is going to be about my predictions, and I will go through how to answer exam questions. So let's go through the first question that we have here. It is important to prevent contamination when growing microorganisms. The diagram shows the transfer and culturing of microorganisms. Name the apparatus labeled A in stage W. So this is apparatus A over here, which I've just highlighted. That is going to be the inoculating loop. So you have to draw a ring around the correct answer. Okay, now the next question is asking you, give the letters of the two stages out of these letters here, which is V, W, X, Y, and Z, which are carried out to kill microorganisms. So which two stages are carried out to kill microorganisms? So the first stage we already know is going to be A, where we are basically passing the inoculating loop through a Bunsen flame, which you can see over here. And then the second one is going to be a Petri dish with agar is heated to 150 degrees for 50 minutes. So it's going to be these two stages here, which is going to be V and W. So I'm going to write this down here. Okay, next one. Give the letter of the stage out of all of these letters here again, which is V, W, X, Y, or Z, where incubation takes place. Incubation is going to take place in stage Z over here, where the Petri dish is kept at 25 degrees for 48 hours. Okay, so that's going to be your answer for this question here. Okay, a culture medium used for growing microorganism contains various nutrients. So microorganisms need nutrients in order to help them survive. Which nutrient is the main source of energy for the microorganism? Draw a ring around one answer, and it's going to be carbohydrates. A lot of students do tend to write down mineral ions, but the correct answer is carbohydrates. Let's move on to the next question. Now, what I've already done, I've already got the answers written down, and it just saves time, to be honest, but I will go through the questions with you. The diagram shows a method used to grow pure cultures of a bacterium. Name apparatus A and B. A, again, is going to be the inoculating loop, and B is going to be the Petri dish, okay? And you are allowed to write down agar plate. That is accepted as well. If you write down Petri dish or agar plate, you will get the mark, okay? Right, let's move on to BI. Why should apparatus A and apparatus B be sterilized before they are used? And the reason for that is to kill any unwanted bacteria. You can also write down to kill unwanted microorganisms. You can also write down microbes, and you can also write down fungi. You can't write down viruses and germs. I just want to make that very clear. You are not allowed to write, for this question here, BI, viruses and germs, okay? Right, let's move on to the next question, which is going to be II. How should apparatus A be sterilized? Apparatus A is the inoculating loop, and we're just going to sterilize that by using a flame. You can write down using a blue flame. I don't think you need to write down blue, but you can just write down using a flame, and that will get you the mark, okay? So this is the question that I've just gone through with you. I'm just going to highlight which ones I've done. Right, let's go through BIII. Adhesive tape is used to secure the lid on apparatus B, which is over here. This is the Petri dish over here. Give one reason why the lid of apparatus B should be securely taped in place. The only reason is because so that bacteria do not get out, and that will get you 1 mark. What is the maximum temperature that should be used in schools to grow the bacteria in apparatus B? That is going to be 25 degrees Celsius, okay? Now, bacteria do grow better at 37 degrees Celsius, but in schools we are only allowed to use 25. If we use anything over than 25, then we can grow very harmful bacteria that can harm us. So that will get you 1 mark. Okay, let's move on to the next question. Right, so a student tried to grow some bacteria in a laboratory. The diagram shows some of the apparatus used. We have the inoculating loop, we have the Petri dish containing the agar, and bottle containing the bacteria. Now here we have the full method. So I'm going to read out the method to you. Now, this is the method that we use. Remove the lid of the Petri dish. Remove the lid of the bottle containing the bacteria. Use the inoculating loop to remove some of the bacteria from the bottle. Spread the bacteria over the agar using the inoculating loop. Put the lid back on the Petri dish. Put the Petri dish into an incubator at 25 degrees Celsius for 24 hours. Steps 1 and 5 could cause the sample of the bacteria on the Petri dish to be contaminated. Give 3 improvements to the method to prevent contamination. So the first one would be to secure the lid with adhesive tape. Pass the inoculating loop in a flame. That's one of the most obvious ones. And you can also write down lift and replace the lid as quickly as possible. Okay, so you don't want any unwanted bacteria to go inside. You can also write down, this is going to get you 3 marks, but you can also write down sterilize agar, and you can write down sterilize the Petri dish, disinfect the bench, and also minimize the exposure of the agar. And yeah, those are the other possible answers that you can write down as well. But you can also write down minimize exposure of agar to the air, dip loop into ethanol after flaming. So these points are allowed. I'm so sorry I've not written it down. I hope you've captured what I've just said. These are the three obvious answers that I've written down, but the ones that I've mentioned, you can also write those down as well. Right, next one, D. Why did the student grow the bacteria at 25 degrees rather than at 40? Tick 1 box. And the main answer for this one is going to be to prevent the growth of harmful pathogens. If we do use anything more than 25, we do have that risk of growing harmful pathogens. Okay, so this is it for culturing bacteria. And these are just previous past paper questions for AQA that I've gone through with you. I really hope that you have found this very useful. The next topic that I am going to go through is going to be to do with plants. So I will be switching over to plants right now. And again, I'll have the answers already written down, and I will explain the answers to you as well. Okay, so thank you so much for watching this part of the video. Let's move on to plants, and that is one of my predictions for the biology paper, which is triple. Sorry, I forgot to mention this is for triple biology, not combined. So this video is mainly based for triple biology. Okay, let's move on to plants. Hello there, so let's go through the topic plants, and I'm going to go through some exam questions. So let's do question 1. The diagram below shows a cross-section of a plant root. The transport tissues are labelled, so one of them is already labelled, which is going to be the phloem tissue, which is over here. And then we need to know what is going to be A, and that's going to be the xylem tissue, okay? Next question, which is going to be AII. Name 2 substances transported by tissue A. You have to write down water or minerals. You can write down ions as well, but you're not allowed to write down nutrients. A lot of students for this question here, which is AII, they write down nutrients and they do not get the mark. So please do not write down nutrients. Write down water and minerals, just to keep it very simple. Or you could write down a named mineral, or you can write down ions, but you need to ignore nutrients, okay? Right, let's move on to B. Phloem is involved in a process that's called translocation. What is translocation? Let's keep this very simple. Right, translocation is the movement of dissolved sugars. You don't have to write down dissolved, you can just write down movement of sugars. You can also write down basically you can make reference to amino acids and sucrose, glucose. You could allow the movement of nutrients, but to be honest, the most obvious answer is going to be the movement of dissolved sugars, and that will get you that 1 mark, and it is the easiest way to remember what is translocation. Right, let's go through BII. Explain why translocation is important to plants. So the reason it's very important is because sugars are made in the leaves. Now remember, photosynthesis is carried out in the leaves, okay? And in the leaves we make sugars, which is glucose. So they need to be moved to other parts of the plant for example, respiration. Respiration uses glucose, okay? So that's going to get you 2 marks over here, and that is the reason why translocation is important to plants, because sugars are made in the leaves, so they need to be moved to other parts of the plant for processes like respiration, or you could write down growth or storage, but I'd write down respiration, okay? And that will get you 2 marks. Right, let's move on to the next question, which is going to be CI. Plants must use active transport to move some substances from the soil into root hair cells. Active transport needs energy. Which part of the cell releases most of this energy? So that is going to be the mitochondria. Okay, so that's where it releases most of the energy. Now, a lot of students really struggle with active transport. Now, what you need to understand about active transport, it's going against the concentration gradient. Now, when it's going against the concentration gradient, it means it's going from, let me show you here. So it's going from low to high. So that is against the concentration gradient. When it's going down the concentration gradient, it goes from high to low, which is osmosis and diffusion. However, active transport is going against the concentration gradient, which is low to high. So explain why active transport is necessary in root hair cells. It's because it's the movement of mineral ions. We need active transport to move mineral ions into the root hair cells, and it's against their concentration gradient, and that will get you 2 marks, and that is my model answer for this question. And a lot of students do tend to write these points down. Okay, any other possible answers could be no, this is the only answer that you can write down. I'll be honest with you, this is what would only be accepted on the mark scheme. So movement of mineral ions against their concentration grade. Now what you need to understand, active transport is basically a process that works by needing energy. So it requires cellular energy, and that comes from respiration. It cannot work without energy. So it's very important for respiration to happen as well. Okay, now active transport is the movement of molecules across the membrane from a region of a lower concentration, like I showed you, to a region of a high concentration against the concentration gradient, and it often assisted by enzymes and requires energy. Okay, let's move on to the next question, question 2. Now the image below shows some cells on the lower surface of the leaf. So we can see where the stomata is labeled. The stomata is the tiny holes that allow gases in and out, but we need to label X. So X is going to be the guard cells, okay? The guard cells open up the stomata, and the stomata is a tiny hole that allow gases in and out, okay? Let's go through B. Water loss by evaporation from leaves is called transpiration. A student set up an experiment to investigate water loss from leaves. A student took 2 leaves, A and B, from a plant, put Vaseline grease on both sides of the leaf, did nothing to leaf A, and wrote down the mass of each leaf, attached the leaves onto a string, as we can see over here, shown in a diagram below. left the leaves for 48 hours, wrote down the mass of each leaf again, calculate the percentage change in mass for each leaf, and give one variable that the student controlled in this investigation. So I wrote down the plants or the length of time. Those are the two variables I'd write down that they have control. Now, the control variable is what you keep the same, okay? So it tells you in the method that it left the leaves for 48 hours. That's the control variable. That's the length of time. And it also says that they basically wrote down the mass of each leaf and then wrote down the mass of each leaf again. So we can see that basically that's where it goes under plants, obviously. Okay, they took 2 leaves. Right, so we're going to move on to the next question over here. Just remember that a control variable is what you keep the same to keep it a fair test, okay? Right, let's go through II, which is here. The mass of leaf A was 1.60 grams at the start of the investigation. After 40 hours, it was 1.28 grams. Calculate the percentage decrease in mass over 48 hours. So when you do the percentage decrease, we can see that the initial mass at the start was 1.6, and then we take away 1.28 from 1.6, because after 48 hours it was 1.28, and then whatever value you get, you divide it by the initial value, which is 1.6, and you multiply it by 100, and that's going to give you 20%, and that will get you 2 marks. That is how you would calculate the percentage decrease in mass. I'm sure it's the same in maths as well as to how you do it. Okay, right. Vaseline blocks the stomata. Remember, the stomata is a tiny hole that allow gases in and out, and we've placed Vaseline to block the stomata for that reason. The percentage change in mass of leaf B was less than leaf A after 48 hours, and explain why. Only because there was less water loss, because obviously we use Vaseline that blocked the stomata, hence why there's less water loss, and that will get you 1 mark. Okay. And let's move on to the next question over here. Give 3 environmental conditions that would increase transpiration. I'll put down heat, low humidity, and wind, and that will get you 3 marks. This question is about plant transport system. Describe how water is transported from the soil to the atmosphere through a plant. Okay, so we know that the process by which how water gets into the roots is going to be osmosis. Okay, so the first thing that I write down is absorbed by osmosis through root hair cells. Okay, osmosis only involves the movement of water. Now it travels through xylem to the leaves. The xylem, remember, it transports water and minerals to the leaves, but we're going to write down travels through xylem to the leaves, which is the water, and it's going to be lost through stomata to the atmosphere by evaporation. This will get you the full 4 marks. So describe how water is transported from the soil. We've mentioned it's transported via osmosis, and then obviously it travels through the xylem to the leaves, and then the water is lost through the stomata to the atmosphere by evaporation. Okay, and that's all you need to write down to get the 4 marks. Right, now let's go into question 3b. Dissolved sugars are moved through a plant in phloem tissue. What is the name of the process that moves dissolved sugars through phloem tissue? And again, that's going to be translocation. Okay, I'm going to move on to the next question over here, which is going to be this one here. Right, this one is based on a practical. So a student used a potometer to investigate the rate of water uptake in a plant shoot. Now, a potometer is a device used to measure the rate of water uptake in plants, which is closely related to the rate of transpiration. Okay, now figure 1 shows the potometer, which you can see over here, this whole diagram that we have. As the shoot takes in water, the air bubble moves. The rate of water uptake in the distance is the distance the air bubble moves in a given time. This is the method that's used. Place the potometer in moist air at 25 degrees. So it's not dry air, it's going to be moist air. Position the air bubble at 0 millimeters in the capillary tube. This is the capillary tube going across horizontally over here. So Number 3, record the position of the air bubble in the capillary tube every minute for 5 minutes. Repeat steps 2 and 3 with the pottometer in different conditions. Table 1 shows the conditions that we used. Over here, we've got A, B, and C, so 3 investigations. And here we had different conditions, such as different temperatures that we have used. Okay, so after investigation A, the air bubble had moved part way along the capillary tube. Suggest how the student moved the air bubble back to 0 millimeters for the start of the investigation B. They would have opened and closed the tap. You can see the tap over here. They would have opened and closed the tap in order to do that. Okay. Capillary tubing is very narrow. Explain why narrow tubing was used. The air bubble moves further along, and then so resolution is improved, so it's a bit more accurate. Okay. So that's the reason why the capillary tubing is very narrow, which you can see over here. It is very narrow so that the bubbles, obviously, so that they can move further along. Right, figure 2 over here shows the results for investigation A. Now you need to calculate the rate of water uptake for this investigation. A in millimeters cubed per minute. Now you can see I've already got the answers here, but I'm going to go through it with you. Now, because it wants you to work out the rate of water uptake for investigation A in millimeters cubed per minute, we have to use number 1, this first equation over here. So what we do, we will be working out the volume. We have to do the distance moved, which is the distance moved by the air bubble in millimeters, which is here. We're going to multiply that by the cross-sectional area, which we already got, which is 0.8 millimeters squared. So the distance moved is going to be 66. You can read that from the graph over here. 66. I'm going to multiply that by 0.8, and that's going to give me 52.8 millimeters cubed. So now we've got the volume, and that's how you work out the volume using this equation here. Now, the second step that we're going to do is work out the rate of the water uptake. So all we're going to do here is the volume, which is 52.8, divided by 5, because you can see the gradient for the graph here is at 5 hours, and that's going to give you an answer of 10.56, and that is your 3 marks. That is how you would do this calculation. I actually have this calculation on my TikTok. I've explained it the same way as how I've done it over here. Right, this is all I'm going to go through in terms of plants. I could possibly go through more, but it's just I have a lot of other topics that I want to go through with you. The next one is going to be based on exercise, and I do advise you, do not just rely on predictions. Remember, predictions may or may not come up. You need to learn everything, okay? So I'm very transparent, very honest. I do tell students that they have to learn everything prior to their exam, not to just rely on predictions. This is just past paper exam practice that I've gone through with you, and I've gone through all the answers with you, and I've tried my best to explain it. I hope you found it very useful. Some of the questions were not necessary that I had to go through, but I really hope you have found this useful. And the next video is going to be based on exercise, and that's one of my predicted topics for biology paper 1. So let's move on to exercise. Hello everyone, I'm going to go through some exam questions based on response to exercise, and this is for module B4 biology. Let's go through question 1. Some students investigated how exercise affects the heart rate. The figure below shows their results. What was student B's resting heart rate? So student B is over here. This is student B. And we can see that the resting heart rate is when they are not exercising. And that is going to be over here, and it's going to be 66, okay? And we can see that the line is completely flat. It goes across horizontally. It's not getting steeper like it does at 2 minutes. So that would be your resting heart rate on the graph. Okay, let's move on to the next question. The student started running at 2 minutes. What evidence for this is in the figure above? We can see that at 2 minutes over here, that the line is getting a bit more steeper, and it's much more steeper for student A in comparison to B. But now we know that they are exercising because the heart rate is increasing. So the heart rate increased, and that will get you 1 mark. For how many minutes did the students run? So we can see on the graph when the heart rate is increasing, that means that they are exercising, they are running. So as soon as the heart rate starts to decrease, which we can see over here and over here, that means they've stopped running. So they were basically running for 4 minutes, between 2 to 6, that's going to be 4 minutes. Okay. Let me just write that down over here. Okay, right. Next question, which is going to be over here. Student B is fitter than student A. Use the figure above to give 2 pieces of evidence that support this statement. So we know that the resting heart rate is lower for student B because it's going to be 66 for student B, and for student A it's going to be 70. And also the heart rate did not increase as much for student B, but we can see that the heart rate did increase much more for student A, and that's your 2 pieces of evidence. And I think this will get you probably 2 marks, okay? That'll be 2 marks. Right, the next question is a 4-marker one. There are other changes in the body during exercise. Explain why these changes occur. So we do need to elaborate in a bit more detail. So we have to state a change that happens in the body during exercise, and we need to explain why these changes occur as well. So the first one is going to be your breathing rate. Your breathing rate increases to provide more oxygen. Okay, so that is the reason why this change occurs. The second one is going to be deeper breathing. So when you're breathing very deeply, it means that you are removing carbon dioxide at a faster rate. So you have deeper breathing to remove carbon dioxide. Another thing that I wanted to mention over here was body temperature increases as more energy is required. And then the next one, which is number 4, sweating occurs, so increased respiration. So sweating can indicate an increase in respiration, particularly during exercise or when exposed to heat. This will get you 4 marks. And in this question, they asked for the change and you have to explain each change, which I have done over here. Okay, so let's go on to question 2. It's quite a long question. During exercise, the heart beats faster and with greater force. The heart rate is the number of times the heart beats each minute. The volume of blood that travels out to the heart each time the heart beats is called the stroke volume. In an investigation, person 1 and person 2 run as fast as they could for 1 minute. Scientists measure the heart rates and stroke volume of person 1 and person 2 at rest. That's when they were not exercising, and then during the exercise and after the exercise. The graph below shows the scientists' results. So here, the first graph that we have here is the heart rate. So this one is going to be I'm just going to get my highlighter on. This is the heart rate, and then the next one is basically the stroke volume represented by the green. Okay, I would really advise you to take highlighters into the exam. It really does help. Right, so let's get started with the question. So the cardiac output is the volume of blood sent from the heart to the muscles each minute. So just gonna get my highlighter out again. I'm going to show you what I'm going to do. So this is my heart rate over here. I'm just going to highlight that yellow. And that's my stroke volume over here. Just one moment. Oops, sorry. Sorry for that. Let me just get rid of it over here. Right, okay. So we need to do a calculation now. The only problem is that the question is on the other page, but I will come back to this page. At the end of the exercise, person 1's cardiac output was 160 multiplied by 77. So 160 was the heart rate, 77 was the stroke volume, which we can see over here with this equation that we need to use. So we need to use the information from the figure above to complete the following calculation of person 2's cardiac output at the end of the exercise. So let's work out the heart rate for person 2, and I'm going to show you how to do this. Now, the person 2's heart rate that we're going to work out on this first graph is going to be 148, and I'm going to show you how I work that out. Now, just one moment. So this is person 2, which is the solid line, and here we can see when they are exercising, it is going to be 148. Now, please make sure that you only look in the area where it says exercise on the graph. I will show you that example in the next graph, what I actually mean. So it's basically stated on the graph where it is at rest and where they are exercising. And we can see clearly over here that is 148, okay? Now, that is going to be the heart rate for person 2. We need to work out the stroke volume. So we need to look at the area where they are exercising for person 2. Now, because we can see that the line peaks over here, but we are only looking at the area where they are exercising, and that's going to be this area where it's grey over here where it states exercise. Let me show you. Can you see that there? So where they're exercising. Okay, so this is the area that we're going to look at. So the stroke volume for person 2 is going to be 38. It's going to be over here. Okay, so that's going to be 38. So the value here was 148, and this one's going to be 38. So I've got this information from the graph, and now I'm going to do the calculation. So heart rate was 148. Stroke volume was 38. So we do 148 multiplied by 38, and that's going to give us 5,624, and that's going to be your 3 marks. So you just have to use the graph to get the information. When I did this exam question with my students, they weren't getting the right results because they weren't looking at the area where they're exercising. They looked at the area where they were not exercising. So just look at the area where they are exercising on the graph. Okay, and let's do B. Person 2 had a much lower cardiac output than person 1. Use the information figure above to suggest the main reason for the lower cardiac output. So the cardiac output is what we just worked out now, which you can see here. And it says use the information figure above to suggest the main reason for the lower cardiac output. And I'm going to write down that they had a lower stroke volume. Okay, so their stroke volume was much more lower compared to person 1. Their stroke volume was actually 77, however theirs was 38. So I'm going to write down lower stroke volume, and that will get you 1 mark. Okay, then that last question that we have here is worth 5 marks, and I've already written the answer down for you because I knew it would take me quite a while to write it down. Person 1, let me read out the question. Person 1 was able to run much more faster than person 2. Use the information from the figure above and your own knowledge to explain why. So we want to know why person 1 was able to run much faster than person 2. So let's look at person 1. Let's see over there. So we can see that they're running much more faster than person 2, which is clearly evident on the graph. So we're going to look at this one over here. It clearly shows that they have been. The solid line is going to be person 2, and the one that's a dotted line is person 1. But we need to know why they're running faster. That's the question it's asking us. So person 1 sends more oxygen to the muscle, to the body, to the lungs. Okay, that will be one of the reasons, which supplies more oxygen. And obviously, when we are supplying more oxygen, we're going to get more glucose. Because remember, oxygen is used for respiration. We need oxygen in respiration. And then obviously that will supply more glucose. Therefore, a faster rate of respiration occurs, and then we remove more carbon dioxide from the body. And that is going to get you a full-on 5 marks, okay? So person 1 sends more oxygen to the muscle, the body, and the lungs, which supplies more oxygen and supplies more glucose, and therefore these two reactants, which are oxygen and glucose, are used for respiration, which is why a respiration occurs at a faster rate, because we've got more of it. We've got more of glucose and we've got more of oxygen. And then because respiration is being taken place at a faster rate, we can remove oxygen more. Sorry, we can remove carbon dioxide more, which you can see over here. Okay, so this is just a practice exam question that I've gone through with you, and it's on exercise. And I'm going to go through my next set of questions, which is going to be on eukaryotic and prokaryotic cells. I hope you have found this useful. I think you are going to get a lot of graphs in your exam where you have to interpret them. That's how most biology questions are. But don't worry, don't get too put off about it. Just know exactly how you need to interpret a graph and what values you need. So I've gone through a practice question with you to help you. Okay, everyone, I'm going to move on to the next topic, which is going to come up now shortly. Okay, everyone, so this video is going to be based on plants, root hair cells, and prokaryotic and eukaryotic cells. I know I said previously it's going to be prokaryotic and eukaryotic. Right, so let's go through some exam questions. Right, 1.1, complete the word equation for photosynthesis. I'll tell you one thing, a lot of students get this wrong, and you might be thinking it's such an easy question, but they do tend to get it wrong. But just remember, we need carbon dioxide plus water, and that gives us glucose and oxygen. And another thing that I've noticed what students do, they write down the balance symbol equation when it clearly says complete the word equation. So don't go through all that stress of writing a balance symbol equation. All you have to do is write down the word equation. Very simple and an easy 2 marks. Right. 1.2, describe how energy for the photosynthesis reaction is gained by plants. So light is absorbed by chlorophyll. Chlorophyll is the green pigment that's found in the chloroplast, and it absorbs the light energy. You can also write down light is captured by chlorophyll or chloroplast. You are allowed to write that down as well, but I've wrote this down because it's just really simple. So that's 2 marks over here and 2 marks here. Okay, so students investigated the effect of temperature on the rate of photosynthesis. The students shone light from a lamp onto pondweed and measured the volume of the oxygen produced per hour. Table 1 shows the results. So here we have the results over here. Just have a look at them. So it's asking you calculate the mean value X. Okay, so the answer is going to be 19.1. So I've added 18.5 plus 19.3 plus 19.5 and divided it by 3, and that gave me 19.1, and that is going to be your mean value for X, which is going to be here. So that's 19.1. Okay, next question. Do students identify one anomalous result? Now, an anomalous result, it does not fit the pattern. Okay, so it's a result that does not fit the pattern. So you need to draw a ring around the anomalous result in table 1. So let's have a look at the different temperatures. So we're going across horizontally. Which one is going to be the anomalous result? And it's going to be this one over here, the obvious one for temperature 45. It's going to be 14.2. We can see it's not really close to 1.9 and 2.2. Therefore, that is the anomalous result. And you just have to draw a ring around it. If you write it over here, that's fine as well. I'm sure the examiner would be okay with that. Sorry, let me just write that down. Okay, and that's 1 mark. Okay, suggest one possible cause of the anomalous result. One of the most obvious reasons could be the value was misread, or you can write down the scale was misread. You can write down there was oxygen in the syringe, the measuring cylinder, the light was moved, temperature change, had different mass, length of pondweed. Quite a few things that you can write down. You cannot write down human error. You can't refer to counting bubbles or time. You can't write down, you can't write down different bulb or different bulb or lamp. The ones that I've mentioned to you are only the ones that you can write down. And I've just wrote down the most easiest one, which is the value was misread, and that will get you 1 mark. Okay, how did the students deal with the anomalous result, which is for 14.2? So they didn't include it in the average. because you can see the average is 2.1. So they did not use it in the calculation or the average. That will get you 1 mark. 1.7, give one factor the student should have kept constant in the investigation. So that's like a control variable. So I've wrote down light, but you can also write down carbon dioxide, concentration, pondweed size, or pondweed species. You are allowed to write down the same piece of pondweed. You are allowed to write down distance, power, color of lamp, light. We cannot accept temperature. The reason we cannot accept temperature is because we are measuring the temperature. That is the limiting factor that we're looking at, which is temperature, which is why you can't mention temperature. So that will get you 1 mark over here. Okay, let's go on to the next question. Table 1 is repeated below. That was 19.1. Why did the rate of photosynthesis decrease from 35, which is over here, to 45 degrees? So we can see that the rate was decreasing. We can see it went from 40.8 to 2.1, which we can see over here. At 30 degrees, it was 44.0, and then after 30 degrees, we can see that it started to decrease the rate of photosynthesis. So I would say that the optimum temperature was 30. I'm just going to write that down over here. So this one over here is the optimum temperature, the ideal temperature. However, after 35, we can see that the rate was basically decreasing. Now, that would be because the enzyme denatured. And what we mean by denatured is that the enzyme is destroyed. So you are allowed to write down that the enzyme is destroyed, damaged. You cannot write down the enzyme is killed. You cannot write that down. You can write down the enzyme loses the shape of the active site. That's allowed as well. Okay, I've just put down enzyme denature as it's the most easiest to remember, to be honest, but please do not write down enzyme was killed. They won't accept that. Okay, so we had to plot the results over here. So it's asking you to label the y-axis. You have to do the scale for the y-axis, and this is the rate of photosynthesis in centimeter cube. You need to put the units there as well, which I'm doing, in centimeter cube per hour. And here's the temperature. This is my scale, just on a basic graph, and this is what your graph would look like. So you have to plot the points onto the graph. Now this is what a graph would look like for temperature, and you can clearly see over here where it's 30 degrees. I did tell you at 30 degrees, that was the optimum temperature where it reaches its peak. So the optimum temperature was at 30 degrees and it was 44.0. And what you can notice after 30 degrees over here, that the rate starts to decrease. So we can clearly see that's evident on the graph. So this is the optimum temperature over here. I'm just going to use a different color. And then after the optimum temperature, we can see that the rate is decreasing after that because the enzyme has denatured. Okay, I hope this has really helped you. I'm just going to move on to the next question, 2.1. What is meant by the term diffusion? Please keep it very simple. Movement of particles from an area of a high concentration to a low concentration. You don't have to make your answer so complicated. This is just an easy answer that you can write down. You can write down the spreading out of molecules. You can write down molecules and particles. You can also write down, you can name some substances, and you can write down, allow down the concentration gradient. You can, those are the only possible points, but this is the most basic answer that you can remember. The answers that I've written down are just the easiest for you to write down according to the question, okay? So that will get you 2 marks here. Okay, let's do this one over here. Figure 2 shows a leaf. Molecules of carbon dioxide diffuse from the air into the mesophyll cells. Which 2 changes will increase the rate at which carbon dioxide diffuses into the mesophyll cells? So increased carbon dioxide concentration in the air. That's going to be one of them. And increased number of stomata that are open. Now, the guard cells open up the stomata. The stomata are the tiny holes that allow gases in and out. Okay, so that will get you 2 marks over here. Remember, it's asking you which 2 changes will increase the rate at which carbon dioxide diffuses into the mesophyll cells. And that's going to be these 2 over here. Okay, let's move on to the next part. 2.4, figure 4 shows a root hair cell. This is a root hair cell. It says name the process by which water molecules enter the root hair cell, and that is going to be osmosis. Now, just remember, osmosis, it only involves water, okay? Next one, nitrate ions need a different method of transport into the root hair cell. Explain how the nitrate ions in figure 4 are transported into the root hair cell. Use information from figure 4. So where it says use information from figure 4 in your answer, please use the information. There's a reason why they're asking you to do that. So firstly, the name of the process is going to be active transport. Okay, this is my favorite topic. Right, so active transport. Now, these are the nitrate ions, which are represented by the X. I'm just going to circle it. The nitrate ions, so we can see the nitrate ions in the soil is a low concentration. So there's a low concentration of nitrate ions in the soil in comparison to in the root hair cell. So it's going from an area of a low concentration to a high concentration, and that's against the concentration gradient. Now, the name of the process is active transport because energy is needed to move. So we need energy to move the nitrate ions from a low concentration in the soil to a high concentration in the root hair cell. And where does that energy come from? It comes from respiration. Respiration is needed for active transport to occur. If there is no respiration, it cannot occur. Okay, so this is your answer for 2.5. And remember, it's asking you explain how the nitrate ions in figure 4 are transported into the root hair cell. And you can mention respiration, that's perfectly fine, energy from respiration, but please state energy in your answer. You can write down to move nitrate ions against the concentration gradient. You can write down there is a lower concentration of nitrate ions in the soil because there is a higher concentration of nitrate ions in the root hair cell. You can't write down atoms and molecules. You need to refer to nitrate ions when you're writing your answer down, okay? But this, again, is the most obvious answer that you can write down. Okay, right, let's move on to prokaryotic and eukaryotic cells. Now, figure 5 shows 3 types of cells. So here we have a bacteria cell, which is an example of a prokaryotic cell. The liver cell is a eukaryotic cell. Okay, not many students know that. The mesophyll cell is a plant cell, but it's also classed as a eukaryotic cell. So plant and animal cells are eukaryotic. This bacteria cell is an example of a prokaryotic cell. So what I'm going to do, I'm just going to draw a little key over here so we can just distinguish between the two. Sorry for that sound of my pen. And then this one over here is oh god, hold on, let me use a different color. I'm going to use purple for this one over here. Just one moment. This one is prokaryotic. Sorry for my handwriting. Okay, right, so give 2 similarities between the prokaryotic cell and the eukaryotic cell. Okay, so you can look at, you are allowed to use the figure 5, the diagram, to help you construct your answer. So we can see that that they both have a cytoplasm. The cytoplasm is over here. That's your cytoplasm where all the chemical reactions happen. And then we can also see that they have DNA, have a strand of DNA in bacteria cell. The DNA is in the nucleus of a liver cell, so it's going to be here in the nucleus, which I've shaded in over here. You can also write down that they both have ribosomes. So there's black dots in each one of them. That is what you call the ribosomes, okay? So make use of these diagrams as well. And you can also write down that let me think what else you could write down. You can't mention anything about the shape for this one. Just go by what I've mentioned over here. Okay, right. 3.2, give 3 differences between a prokaryotic cell and a eukaryotic cell. And again, it says use figure 5. This is where you're going to get your answers. Now, you can use your own knowledge. Now, a lot of students will know that prokaryotic cells have no nucleus. Okay, so they do not have a nucleus, prokaryotic cells. But what I've done is I've not wrote that down over here, but that is one of the most obvious answers that you can write down. But I've wrote down that prokaryotic cell is much more smaller. So we can see it's only 1 micrometer here, and we can see the liver cell is 30 micrometers, and that obviously the mesophyll cell is 0.05 millimeters. So prokaryotic cell is smaller. Okay, I've also wrote down prokaryotic cell has no mitochondria. We can see there's mitochondria. So this is the mitochondria over here, which I've just circled in the mesophyll cell and the liver cell. So we can see that the prokaryotic cell has no mitochondria. And I wrote down that prokaryotic cell have plasmids, which are extra rings of DNA, which I've just circled over here. Those are the plasmids, whereas these two do not, which is the liver cell and the mesophyll cells do not have one. You can also write down prokaryotic cell has no nucleus. That's one of the obvious answers you can write down. You can write down the DNA is free in the cytoplasm, or the genetic material is free in the cytoplasm. You can also write down prokaryotic cell has a single loop of DNA. Prokaryotic cell has plasmids, which I've already mentioned. You can also write it in converse for eukaryotic cells. If you decide to write down eukaryotic cells have a mitochondria, whereas prokaryotic cells do not. So you can do it that way as well. So it's entirely up to you how you want to do it. Okay, as long as you're comparing and you're writing the differences and not the similarities, and make sure that you use the diagram that they give you. If they're asking you to use the information from a diagram, then I would advise you to use it, and also your subject knowledge as well. Right, this is it now for eukaryotic and prokaryotic cells. I've gone through quite a range of different questions. These are just practice past paper exam questions, and I wanted to talk you through how to answer them. So yeah, my next topic is going to be based on monoclonal antibodies and how they're used in pregnancy tests. Hello everyone, I am going to go through a topic that is therapeutic cloning, and this is for biology triple students only. So how does it work? So let's start with step 1. So the body cell is taken with the desired genes that we need. So this here is the body cell, which I've just circled over here in red. That is your body cell, and it's got the desired gene in there that we need. Okay, so the nucleus, you can see now what's happened is that the nucleus has actually been taken out of the body cell. The reason we've taken out that nucleus is because it has that desired gene. So the nucleus is taken out containing the DNA. Step 3 over here, the nucleus is removed from another egg cell and disposed of. And then we can see over here at step 4, that the nucleus that came from the body cell, which has the desired gene, is put into the nucleus of the egg cell, which is over here, okay, which I'm going to circle right over here where we have step 4. Okay, and now the egg is forced to divide into many stem cells. So this is how therapeutic cloning works. Now, why the body cell? Because the body cell can't be turned into any type of cell. Why? The nucleus contains the patient's genetic information. Why the egg cell? The egg cell will divide and form an embryo, but it can't have 2 nuclei or the cell will die. And then why? Now the egg cell has the same genetic information as the patient, allowing the patient's body to accept it. And this is how therapeutic cloning works. So you may be asked in your exam to explain how it works, and this is a step-by-step process. So what I would advise you to do is write down each of the steps from 1 to 5. If you want to use diagrams, that's perfectly fine. It's good to remember using diagrams, but this is what you need to know. Now I'm going to go through a series of exam questions based on therapeutic cloning. Hello there, we are going to go through some exam questions based on therapeutic cloning. So we're looking at this question over here, which is F. Figure 2 shows how embryonic stem cells are produced in therapeutic cloning for the use in patients. So these are embryonic stem cells. And here we have the diagram over here, which it shows how the embryonic stem cells are produced in therapeutic cloning. We can see in this diagram. So this is a donated egg cell. And then we have the body cell from the patient, just the same as the diagram that I went through with you earlier on. And here we have all the cells cloned, okay, where we have the embryo. Now it's asking you give 2 advantages and 2 disadvantages of therapeutic cloning in medical treatments. So the first advantage would be it may be used to cure diseases. That's one of the main advantages. You could write down it could treat diseases, or it could cure them. You can write down it cure medical conditions, produce replacement cells, but I've wrote down the most obvious. Advantage 2 would be cells of any type could be produced. That's the reason why we use embryonic stem cells, for that reason. Disadvantage would be the first one, embryo is destroyed, so that will be related to ethical issues, that it's wrong. Disadvantage 2, do not know the risks and the side effects. So you are also allowed to write down, let me go through all the other possible answers. You can also write down that the embryo is, oh, I've already told you about the embryo. You can write down there is a shortage of donors and eggs. Not many people want to donate eggs. You do not know the side effects. We can also write down we may transfer a viral infection as a disadvantage, poor success rate to produce viable eggs, embryo. So those are the other disadvantages, but I've just wrote down the most easiest ones for you to remember. Right, next question. Question 3. Stem cells can be collected from human embryos and from adult bone marrow. So those are the 2 places where stem cells can be collected from. Stem cells can develop into different types of cells. The table gives information about using 2 types of stem cell to treat patients. So here we have stem cells from human embryos and stem cells from adult bone marrow. Now it's asking you, scientists are planning a new way of treating a disease using stem cells. Use only the information from above to answer these questions. So please make sure you use the information above. Give 3 advantages of using stem cells from embryos instead of from adult bone marrow. So let's look at the human embryos. Let's look at the information that we have. So it costs $5,000 to collect a few cells. I wouldn't say that's an advantage. There are ethical issues in using embryo stem cells. I don't think that is an advantage. The stem cells can develop into most other types of cells, so that is definitely an advantage. Each stem cell divides every 30 minutes. So it's a very quick process. Advantage, and there is a low chance of a patient, let me just see the last one, immune system rejecting the cells. That will be an advantage as well. And more research is needed into the use of stem cells. That would be obviously that wouldn't be an advantage. Okay, so can you see I wrote all these advantages down? It's about you identifying the advantages. Okay, they're not all advantages in that table, which you can see here. They're not all advantages. That you need to identify them, which I've helped you with now. So I wrote down can develop into most other cell types, low chance of rejection, each cell divides every 30 minutes. So that's your 3 marks just from that table. Okay, next one, give 3 advantages of using stem cells from adult bone marrow instead of from embryos. So it costs £1,000 to collect many cells. So we can see it costs $5,000 for a human embryo. So it is much cheaper. You can write that down. Adults give permission for their own bone marrow to be collected. That would definitely be an advantage. The stem cells can develop into only a few types of cell. I wouldn't say that is an advantage. Each stem cell divides every 4 hours. Nope, that takes too long. There is a high chance of a patient's immune system rejecting the cells. That's definitely a disadvantage. And the use of these stem cells is considered to be a very safe procedure, and that is an advantage. Okay, so again, we are identifying them. So I wrote down cheaper, only cost £1,000, can collect many stem cells, safe, and adults give permission for their bone marrow to be collected. You can either write down safe or adults give permission for their bone marrow to be collected, and that's your 3 marks. How easy was that question based on therapeutic cloning? So I've gone through the process of how therapeutic cloning is carried out, and I've done a series of exam questions. Please make sure you learn the advantages and disadvantages of embryonic stem cells and adult stem cells, and the advantages and disadvantages of therapeutic cloning as well. Okay, thank you. Okay, so I'm going to go through monoclonal antibodies. So this is an application of knowledge question, which is using and producing monoclonal antibodies. Now, monoclonal antibodies are used to measure the levels of hormones in the blood. Pregnant women produce the hormone HCG. So I'm going to underline that information over here. HCG is excreted in the urine of a pregnant woman. Figure 1 shows 4 pregnancy test strips over here, and we've got A, B, C, and D. And it tells you which one is a positive result, a negative result, and an invalid result. So a positive test result is when a line appears in the control window and the result window. So this here is a control window, which I've underlined, and this is the result window. So that would mean that the female is pregnant. And then a negative test result is when a line appears only in the control window, which you can see over here. Okay, so which test strip shows a negative test result? So the negative test result is going to be in A. The reason it's going to be A is because a line has only appeared in the control window, which you can see A over here. There's no line in a result window, which would mean it's a negative test result. Monoclonal antibodies are used for pregnancy testing. Give one of the use of monoclonal antibodies. So it can be used to treat some cancers. I'm going to write that down. It could be used to treat some cancers. It could be also used to identify other hormones or locate blood clots. The most obvious one is to treat some cancers, okay? Now, figure 2 shows the parts of a pregnancy test strip, which we can see here. I've already got the answer written down. So we have the control window, the result window, and the reaction zone, and where the urine is applied, okay? Now, what I'm going to do is I've wrote down 5 points here to help you understand how to answer this question. So the pregnancy test strip will show positive test result when a woman is pregnant. Explain how the pregnancy test strip works to show a positive result. So the first thing that you need to write down is that urine passes through the reaction zone, okay? So that is where the urine is applied. It's applied where it says number 1 over here, which I've coloured in red. But then what would happen is that the urine would then pass through the reaction zone, which we have over here. Now, the HCG hormone is found in the urine of a pregnant woman. It tells you here in the question. Pregnant women produce the hormone HCG, which is excreted in the urine. So the HCG hormone is going to bind to the mobile HCG antibody, which is going to be in the reaction zone, which you can see there. The HCG hormone binds to the immobilized HCG antibodies in the reaction zone. The other antibodies, which do not attach to the HCG, bind to antibodies in the control zone, which we have here in number 4. Okay? So the blue dye appears in both control and result zone to show a positive result, and that would mean that the female is pregnant. Okay, so these are the 5 points that you need to write down to get the full 6 marks. Okay, I really hope this has helped you to understand monoclonal antibodies. You may need to know how monoclonal antibodies are made. This is just one of the uses of monoclonal antibodies, which I've gone through with you. So I hope this has helped you, and just take a screenshot of the answers or write them down, but just make sure you do learn all the uses of monoclonal antibodies prior to your exam. Hello there everyone. I'm going to go through a mixture of exam questions, and that are based on my predicted topics that I think may come up in the exam. Now, just bear in mind that these are predictions, they may or may not come up. Please do not just rely on the predictions. It's really important to practice as many past papers, as past papers do help you with your exams, but do make sure that you do not only rely on the predictions. So let's go through the exam questions. Right, 3.1, complete the word equation for photosynthesis. Now, I'll tell you one thing, I see a lot of students write down the symbol equation when really you need to write down the word equation. So just make sure that you do write down the word equation. I think if you write down the symbol equation, you'll still be given the mark, but don't make it too difficult for yourself. So the word equation for photosynthesis is carbon dioxide plus water gives you glucose and oxygen. Right, let's go through this practical question. A student investigated photosynthesis using pondweed. Figure 3 shows the apparatus the student used. This is the method that they have used. Step 1, set up the apparatus as shown in figure 3. Step 2, switch on the lamp. Step 3, after 20 minutes, record the volume of oxygen collected in the measuring cylinder. Step 4, repeat steps 1 to 3 using bulbs of different power output. What was the independent variable in the investigation? So it's going to be the power output of the bulb. The independent variable is what you change, and it clearly states in step 4, repeat steps 1 to 3 using bulbs of different power output. Okay, 3.3, suggest 2 ways the method could be improved so the results would be more valid. These are my 2 possible answers, repeat and calculate a mean. Please, in your exams, do not write repeat on its own. You have to write down repeat and calculate a mean to get 1 mark. Control the water temperature could be one of them, and you can also write down control the concentration of carbon dioxide gas. And you can also write down control the distance of the bulb from the pondweed, control the mass of the pondweed, and repeat and to eliminate the anomalous results. Okay, and you can also write down, I've wrote down control the water temperature, which is fine. Okay, so that will get you 2 marks. Okay, so this calculation question, which is only worth 1 mark, and this is, I think it is let me see which level this question is. An AO2 question. Right, so AO2 questions are like high level questions, and these are the sort of questions that are going to be assessed on the triple biology paper. Right, calculate value X in table 3. So what we're going to do over here, I'm just going to rub this out over here. We can see that the volume of oxygen collected in 20 minutes is centimetre cube, and then the rate of photosynthesis in centimetre cube per hour. So all we have to do is work out how many 20 minutes there are in an hour. So I do 20 multiply by 3 gives me 60 minutes, and that is 1 hour. So what we do with that 1.1 is 1.1 times by 3 is going to give you 3.3, and that's going to be your answer. Okay. Right, so next one over here, I have more or less done already for you. Complete figure 4, so you should label the x-axis, which is going to be the power output of the bulb in watts. That's done, so that'll get me 1 mark. Use a suitable scale, I've gone up in 50s, that is done. Plot the data from table 3. So this is table 3 over here. So you're going to plot the rate of photosynthesis in centimetre cube per hour. And then you're going to draw a line of best fit. So this is my line of best fit that I have drawn already. It's a curved line of best fit. Now it wants you for 3.6 to determine the expected rate of photosynthesis with a bulb of power output of 75 watts. So 75 watts is, I'm just going to zoom in a little bit now. And 75 watts is going to be around about over here. So this is where 75 watts is. And then we draw a line going up vertically. This is how you should ideally do it. This is not going to be the best example. I'm not using a ruler. And then we draw a line going across horizontally. And I think that's going to be 1.8, okay? Which we can see over here. And that is your answer. That's the rate of photosynthesis at 75 watts, and that's going to be 1.8. You can get a range of answers as long as it's between let me check. It can be between it can be 1.8 and 1.9. But it can be 1.8 and 1.9 if no line of best fit or incorrect graph is drawn, okay? Right, but I've got the answer as 1.8, and this is how you would work it out. You need to make sure that you have drawn a line of best fit in order to work it out. It just makes it easier for you, okay? And that's how you would do it. Okay, let's move on to the next question. Which graph shows the effect of temperature on the rate of photosynthesis? And it's going to be the third one over here. It's going to be this graph over here. So where it reaches the peak over here, that's where we have the optimum temperature. Just remember that this part where it goes up, that is your optimum temperature. And then you can see the rate is decreasing. That means the enzymes have been denatured because the temperature is too high. Okay, so I just thought I'd go through that part with you. Right, let's move on to the next question. And again, I've got all my answers written down. It just saves time, to be honest. I didn't want to make the video too long. Right, question 4.1. Water moves from a plant to the atmosphere through the leaves. How is the volume of water lost from the leaves controlled? So how the volume is controlled, how the volume of water is controlled is through the stomata. So it's the opening and closing of the stomata. The guard cells, which is a specialized cell, it opens up the stomata, just to let you know. So that's going to be 1 mark, okay? 4.2, describe the transfer of water through a plant from the roots to the atmosphere. Okay, now we know that water is transported in the xylem vessel. So the xylem vessel transports water and minerals to the leaf of the plant. Okay, but it is not asking us about the minerals, it's only asking about water. So that will get you your first mark. Water evaporates from the leaves, and that will get you a second mark, and it's going to be through the stomata by where the water evaporates. Now for this question over here, you can also write down that water, if you write down water evaporates, that's perfectly fine, but you can also write down loss of water vapor, and you can write down through the stomata or allow between the guard cells. Okay, but what I've written over here is one of the most obvious answers and easiest ones that you could write down. I didn't want to make it too complicated. Okay, sorry, just gonna sort this out over here. Right, let's move on to the next question. Right, a student investigated the volume of water lost from 2 plants of different species. Both plants were kept together. Figure 5 shows the student's results. So here we have plant B and we have plant A over here. It's asking here for 4.3. Suggest one reason for the difference in the rate of water loss from the 2 plants in the first 2.5 hours. So we can see that plant A had a higher volume of water lost in comparison to plant B. Okay, so we can say that the reason is because the plant A has more stomata. That could be the reason why it has a higher volume of water loss. You can also write down that you don't have to, there is a possibility that you can write other answers, such as plant A has more leaves, or you can write down plant A has bigger leaves, or you can write down plant A has a greater total surface area of leaves. But I've wrote down plant A has a stomata, and you can write it in converse for plant B as well. Okay, so I wrote down plant A has more stomata, which is the reason why the volume of water was quite high. That which was over here, you can see that the volume of water loss was quite high for plant A. Okay, right, let's go into 4.4. This is a calculation that we need to do. Both plants were moved to a different place at 2.5 hours. Calculate the rate of water loss per hour in plant B from 2.5 hours to 3 hours. Give your answer to 2 significant figures. Right, so it's just like working out the gradient. So when you're working out the gradient, it's always y over x, okay? Let me just show you here. So gradient equals y divided by x. So we need to work out between 2.5 hours, which is over here. I'm just going to mark it here. This is 2.5 hours and 3 hours over here. Okay, so this is going to be your x-axis and that's your y. So we need to work out the difference in x. So we just do 3 take away 2.5, which is going to give us 0.5. And then we work out the difference in y. So this reading that we have over here is going to be 2, which we can clearly see. So this is going to be 2. And then this one here is going to be 7.2 over there. Okay, so what I'm going to do, I'm just going to show you how I'm going to do this. I'll show you over here, as we can see the graph here. I'll do 7.2 take away, just one moment, 7.2. Just bear with me, let me work it out. I'm just going to do it on my calculator. 7.2, we're going to take away 2 is going to give us 5.2. And then we do 3 take away 2.5 is going to give us 0.5. So all you do is 5.2 divided by 0.5, and that's going to give you an answer of 10.4, and that's going to be your answer. So you round up to 2 significant figures, and that's going to give you 10. Okay, so the answer is going to be 10 for this one. I've done the working out over here, so it's just like how you work out the gradient. Okay, so let's move on to the next question. 4.5. Suggest 2 reasons why the rate of water loss in both plants change after 2.5 hours. The most obvious reason would be it was warmer. So if it was warmer, that would mean light intensity was higher. Okay, and that's for 4.5. Right, I'm just going to quickly go through this question, which is based on mechanical and biological valves. And I've got a 6-mark question here that I want to go through with you as well. So let's go through question 5.2. A patient with a leaking heart valve may have the valve replaced. A study compared 2 different types of replacement heart valve. We have mechanical valves, and then we have biological valves, which come from pigs. The data used in the study was collected from female patients aged 50 and 69. Table 4 shows the data. Now it's asking you in this question, give one conclusion about the death of patients from the heart-related problems after a valve replacement. So it says over here, include calculations to support your answer. So we're going to look at this column over here, which is the number of patients that who died from heart-related problems after valve replacement. Okay, so this is the column that we're going to look at. So all we have to do basically is we need to do one. Let's look at mechanical first over here. So I'm going to write down mechanical. So we know there was a total of 180 patients that died after the replacement. So we do 180 divided by 2852, and then times it by, we're going to times that by 100, and I think that is going to give us something like, let me think, that'll give us about 6.31%. Okay, so that's the first calculation. So let's do the second one, which is for biological. So we're going to do again 178. We're going to divide that by the total, which is 1754, times it by 100, and that's going to give me an answer of 10.14%. Okay, so in conclusion, we can say that the third mark is right down there's a higher proportion of patients that die with a biological valve. Okay, so therefore, let me write that down. So therefore, there is a higher proportion of people that are more likely to die with a biological valve. All right, I'll change that to patients over here. Okay, right, so that will get you 3 marks, okay? Right, now this is a 6-mark question that I'm going to do with you. But the first one, let's do 5.3. One risk of mechanical valves is that blood clots can form on the surface of the valve. Name the component of the blood that starts the process of blood clotting, and that's going to be your platelets, okay? So that's going to be your platelets, and that's 1 mark. Right, evaluate the uses of mechanical replacement heart valves and biological replacement heart valves. You are allowed to use the information from table 4, and you have to use your own knowledge. Now, if you only use the information from this table, you're not going to get more than 2 marks. And believe me, that is the truth. You have to use your own subject knowledge as well. So you could write so much information down, but if you've not used your own knowledge, you're going to probably not get more than 2 marks. And I'm going to be very clear about that. So let me go through what I wrote down. So with mechanical valves, this is how I would set out my answer. I'd separate mechanical valves, biological valves, and then I write down a conclusion. So I wrote down mechanical valves are longer lasting. We can see that it says over here, percentage of patients needing a second valve replacement within 6 years, it's only 2.2%, whereas with biological, it's 5.2%. So I've just wrote down it's longer lasting, less likely to need replacing. Blood clots are more likely to happen. We can see over here, I think it states it over here. It's a 5.8% of patients who have a blood clot on the brain after surgery. And then I've also wrote down patient has to take anti-clotting medication. Some patients say they can hear their valves opening and closing. Survival rate at 5 years is slightly higher for mechanical valve. Okay, these are all the possible points that I've written down for mechanical valves. And most of it is my own subject knowledge, and I am allowed to use information from table 4, but you need to back it up with your own knowledge. Okay, biological valves, no additional medication is required if you get a biological valve. Ethical issues surrounding the use of animal tissue, some people do not agree with it. The valve may harden, which is why it needs to be replaced more often. We can see over here. Percentage of patients needing a second valve replacement within 6 years, because it's 5.2%, and then more likely to be rejected. And then both valves are, they both are readily available, and there's little wait time. So this is my answer to the 6 mark question, and you can see how I've used points from the table over here, and I backed it up with my own subject knowledge as well. And that will get you 6 marks. Okay. Right. What I'm going to do now, I'm going to go through just probably a few more questions, and then I think I am done from there. Okay. Now let's go through this question over here. Pancreatic cancer develops a malignant tumor. Now, a malignant tumor, it is a secondary tumor. Okay. So it is a secondary tumor, meaning it can spread to other parts of your body. So pancreatic cancer develops when a malignant tumor grows inside the pancreas. Now, the pancreas produces digestive enzymes. What is an enzyme? So the best definition that you can write down to get 2 marks is it is a biological catalyst that is a protein. This is the easiest way to remember it, and that will get you 2 marks because enzymes are made out of protein. 8.2. Carbohydrates is an enzyme produced by the pancreas. Name 2 other organs in the digestive system that produce carbohydrates. And so the first one is going to be the salivary glands, and the second is going to be the small intestine. Okay. And then 8.3. One symptom of pancreatic cancer is weight loss. Explain how pancreatic cancer may cause a person to lose weight. Do not refer to hormones in your answer. So please do not write about hormones in your answer. So what I've written down here is that it reduces the enzyme production, because remember, in the pancreas, the pancreas does make enzymes to help digest your food. So it's going to reduce the enzyme production, and for that reason, your food is not going to be broken down fully. Okay, so your food ain't going to be broken down fully, or the food is not digested fully. And then therefore less glucose is absorbed into the bloodstream. So if there's less glucose, that means there's going to be less glucose for respiration to occur. And this will get you 4 marks. Okay, all these points that I've written over here will get you 4 marks. Okay, right. I'm just going to go through one more question now, which is going to be this one over here, which is going to be 8.6. So 8.6, the drug was trialed before it was licensed for use. Sorry, this is part of another question. To improve the validity of results in the trial, some patients were given placebo, which is like a fake drug. and a double-blind trial was used. That's when neither the doctor or the patient knows what drug is being used. Give reasons why a placebo and a double-blind trial were used. So I wrote down to avoid the patients thinking they feel better when they to avoid the patients thinking they feel better with the drug, and for double-blind trial is to avoid bias. So that will get you 2 marks over here. This is to do with this question over here, which is to do with drug trials. Okay, let's go into 8.7. One stage in a drug trial is to test the drug on healthy volunteers. What is the next stage in a drug trial? So the next stage is going to be testing on volunteers that actually have the disease. Okay, and that is 1 mark. Right, this is it for now. I've tried my best to go through as many questions with you, and please do practice lots of past papers. You know, practicing past papers is very essential part of your revision. It's very important. When you're learning all the content, that's great, it's fantastic, but then you need to know how to answer the exam questions. A lot of students struggle, and it's because they don't have enough exam practice. Now, when you do a past paper, you should test yourself. And if you are, when you're doing the past paper, be honest with yourself. And then if you don't get the mark that you obviously, sometimes you may not get the grade that you want, you may get a low mark. Keep practicing again. Practice that same past paper over and over again. Let that information stay in your head, because that's the only way you are going to get better at answering exam questions. The more you practice, the better you be. So when you do it the first time, you're not going to get the best result. But if you keep doing it over and over again, you will get a good outcome out of it. You are putting in all that effort and you are going to get something good out of it. So just carry on practicing and everything. Keep practicing again and again. Right, I'm going to put a video on for chemistry, which will be in a couple of days. But I really hope that this has helped you. Please leave your comments below as to how you found my exam questions and everything, and if there's anything that you want to clarify with me, please leave a comment below and I shall reply to you as soon as I can. Good luck for your biology paper 1 exam. Please make sure you learn everything, and please let me know how you did. Thank you. Hello everyone, I'm going to go through my predicted topic, which is going to be the body defense system, and I'm going to go through this 6-mark question, and I will give you the model answer as well, which you can see it's already written down just to save time. So describe how the human body prevents pathogens from entering, defends itself against pathogens inside the body. So prevents pathogens from entering, we can write down the skin, which acts as a barrier. Scabs form over cuts. Platelets are involved in forming clots. The stomach contains hydrochloric acid, which kills bacteria in our food before there is any contact in our blood. The eyes produce tears. Your tears contain enzymes to kill bacteria. Your breathing system, you can mention your nose. It produces mucus. Mucus is very sticky and it traps bacteria. Mucus is carried away by cilia, which are the tiny hairs. The second bullet point, which is defends itself against pathogens inside the body, we can mention the immune system produces white blood cells. White blood cells engulf pathogens. Antitoxins are produced, which neutralize the toxins. Antibodies are produced which help destroy pathogens. Memory cells are formed, which give a more rapid response if the pathogen re-enters. So if you get that same disease again, your immune system will react much more faster and it'll be a rapid response. So this is the model answer for this exam question. And just remember, it is a predicted topic. It may or may not come up. And I really hope it has been useful in terms of your revision. Thank you.